أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم لا أقسم بهذا البلد وأنت حل بهذا البلد ووالد وما ولد لقد خلقنا الإنسان في كبد أيحسب أن لن يقدر عليه أحد يقول أهلكت مالا لبدا أيحسب أن لم يره أحد ألم نجعل له عينين ولسانا وشفتين صدق الله العلي العظيم. Lecture number 26, and we are studying very useful information. We are studying enzymes are composed of protein. Protein is composed of amino acid. We are studying active site integral part of enzyme. We are studying active site combined with substrate to form ES complex. We are studying allosteric site is inactive site of enzyme. We are studying substrate react with active site to form ES complex, and ES complex change the reactant into product. We are studying protein has four structure: primary, secondary, tertiary, quaternary, and hemoglobin has quaternary structure. Enzymes have quaternary structure, they are more complex. We are studying substrate concentration increase, rate of reaction increase. We are studying temperature is very important. The enzymes work at maximum temperature. That temperature is almost 37 Celsius in human body. That temperature is called optimum temperature. We are studying that when the sequence of amino acid will disturb, that enzymes are denatured. We are studying that the energy which is required to convert the reactant into the product, that is called activation energy. We are studying enzymes work by decreasing activation energy. We are studying that the molecules collide, means substrate collide before reaction, and their collisions, proper collision is called effective collision, and that collision is involved to convert the reactant into product. And during that collision, effective collision, we can say that activation energy is achieved. We can study that we are discussing that activation energy basically involve in creating effective collisions, and we are studying that enzymes active sites can help sometime need help by cofactors, which are of three types: prosthetic group, cofactors, and activators. And activators are not going to attach with active site. We are studying prosthetic group, that cofactor which help the active site, but it will attach with active site with the covalent bond. We are studying that coenzyme will attach with active site but with the weak linkage. And we are studying the enzymes are specific: lipase digest the lipid into fatty acid and glycerol, and similarly the pepsin works in acidic medium. This is my today's discussion, that pH value of enzymes are different and enzymes are specific. They work at the different temperature. If you see these enzymes, you can see, first of all I will tell you, your body system is made like that. Your body system is made like that. In your oral cavity, yani buccal cavity, temperature pH value is the basic. Yeah, this is amazing arrangement of the pH in the different parts of the body. In one part of the machine, pH is basic, then acidic, then basic. Why they are changing the why these pH values are being changed from one part of the body to another part of the body? Reason: nature is allowing to enzymes to work, because all enzymes are not working at one pH. Otherwise your normal pH in the blood is the 7 point something, which is buffered solution. To add 7, it will not work. All enzymes need different pH, so requirements are given to enzymes for proper functioning. Adjusting the creator is adjusting the function of enzymes by changing the pH in the different parts. This is a super arrangement, that at one part of the body pH is different, then another part comes, then pH change, then another part comes, the pH change. I give you example. This is oral cavity, buccal cavity. Amylase enzyme is here, and amylase will digest the starch to glucose. The medium is the basic. Why the medium is basic? Is favoring us? Yes, it's favoring us. It's favoring amylase enzyme. What is the requirement of amylase enzyme? Amylase enzyme will work in a basic medium. That is one of the main reason. Then go to esophagus. Esophagus, there is no enzyme. Comes the stomach. What happens in stomach? What happens in stomach? In stomach, pepsin is present. What is the medium of the pepsin? You studied digestive system in the 9th class. You studied digestive system in the 9th class. Pepsin work in acidic medium. pH change to acid due to HCl secreted by gastric gland. Sir, why? How these pH values are being changed? Like chemical compounds which are present in our body, nature use those chemical compounds in a super fashion. Sir, how these chemical compounds are present in our body? Minerals. Why the doctors are telling you need to take the minerals, take iodine, take sodium, take calcium in the form of vitamins, and take these things? So where these things are present, I can purchase calcium, sodium, iodine from the Kiryana store, from the shop, from cash and carry? No, you cannot purchase, because that is present in the vegetables, that are present in all type of crops. You eat all things, fruits, vegetables and meat, everything consists of these vitamins, minerals. Even our water consists of these minerals. So what happen? Our stomach consists of HCl and pepsin work in the acidic medium. Go back to first part of small intestine, duodenum. What happens? Solution become basic once again. Yeah, this is a super arrangement. This is I will stop here. Just let me tell you, you can make little machine that changing the pH from our themselves. Not you will do. The machine adjusts the pH himself, and according to requirement of enzyme changing the pH. Look here, amylase was there, basic medium created due to sodium bicarbonate NaCO3. Pepsin was there, the basic acidic medium created due to HCl. When trypsinogen by pancreas and trypsinogen by pancreas, the lipase by the pancreas library, amylase by the pancreas, enterokinase by the intestine, when they were here, these enzymes were here, they changed the medium changed to pH. Basic medium reason, because these enzymes work in a basic medium. What is the learning of this knowledge? Learning of this knowledge is that enzymes work at the specific pH. One more point I was discussing, that if the pH value is 1 to 14, I discuss here 1 to 14 pH value, 7 is a neutral, 1 to 7 solution basic, acidic, sorry, 1 to 6, because 7 is a neutral, from 8 to 14 basic. This is strong acid, this is a medium, this is a strong acid, medium acid, weak acid because it is closer to neutral, weak base, 8 medium base, and the strong base. Now if you are telling that 1 to 6 enzyme is working, mean acidic medium is working. I give one example, I think so, pepsin. So we see the pepsin value. What is the value? I told 2. I was just guessing that it is closer to 2, but I was not confirming you. Now you can see 2 value of the pH. What is the pH? 2. Why it is written optimum pH? Definition of optimum pH: the pH value at which the enzymes work maximum, that value is called optimum pH. The pH value at which the enzymes work maximum, that value is called optimum pH. So if you are talking generally, this is very important point, you should write this point when I will explain. You must possess pencil and ballpoint and copy. If any information which is a much precious, you must write. You should make the notes by yourself, just you made in classroom, my dear students. If you see 1 to 6 is acidic medium. Significant point I am telling, be alert, be attentive please. 1 to 6 is acidic medium, but here what is written? Pepsin work in 2 pH. So it means that pepsin will work 2 pH maximum. So this pH is optimum pH. Normally we say, I am asking question to you. Normally we say pepsin work in acidic medium, mean that pepsin can work between 1 and 6, mean that pepsin can work in 1, pepsin can work in 2, pepsin can work in 3, 4, 5, 6. Answer is, question is that if optimum pH of pepsin is 2, so is pepsin can work in other pH which is acidic medium, because normally we say pepsin work in acidic medium. If pepsin cannot work in 4 value, 5 value, 6 value, then why we are telling pepsin work in acidic medium? We should not tell pepsin work in acidic medium. We should not tell pepsin work in acidic medium, because reason is that if you are telling that pepsin work in acidic medium and you are confirming that pepsin can work in all mediums, so in the same time you are telling pepsin work in 2 medium, so you must say pepsin work at 2 pH. You must say pepsin do not work at 3 pH. We are telling acidic medium, it means pepsin is working in all these mediums. Yes, this is right. Pepsin is working in all these medium of acidic range, mean 1 to 6. 1 to 6. 1 to 6. So 1 to 6 is the range. 1 to 6 is the range in which pepsin is working. So when we are talking about pepsin work in acidic medium, it means that pepsin is ranging from 1 to 6. So why 2 is written then? If pepsin can work at 1, pepsin can work as 2, 3, 4, 5, 6, in all these mathematical values pepsin can work, because we are telling pepsin work in acidic medium. Why 2 we say, why 2 is optimum temperature? This is good point. Now I am disclosing the answer. 2 mean acidic medium mean the pepsin will work in all ranges of pH value 1 to 6, which is the range of acid. Clear? Because 1 to 6 solution is acidic. But 2 mean at 2 the pepsin will work maximum. Clear? Pepsin will work, each enzyme has a specific pH at which it works maximum like temperature, and that pH is called optimum pH, and that temperature is also called optimum temperature. We'll go back to the discussing some pH. Pepsin work in 2 optimum pH. He did not tell optimum, he did not tell the pH. Why he did not tell the pH? Because pH he cannot tell. pH he cannot tell. He has to mention optimum pH. So there are 2 ways to explain the enzymes. One way I tell you by watching this graph, there are 2 ways. One way is that I say pepsin, whenever solution is below 7, the solution is acidic. When the solution has more than 7, that is the basic. So I can explain by simple way by using the name acidic and the basic. For example, I name pepsin is acidic. Pepsin work in acidic medium. Sucrase work in acidic medium. Enterokinase work in almost less acid, weak acid medium. Salivary amylase just little neutral medium. Catalase just little basic medium. Lipase in basic medium. Arginase enzyme present in the DNA in the basic medium. So the point I just wanted to explain, these details are simple details. Normally we say amylase work in basic medium, pepsin work in acidic medium, lipase work in a basic medium. These pH values are confirming that they are working at maximum. They are working maximum at this pH. They are working maximum at this pH. So these things are telling, these pH values are confirming that enzymes work maximum. So we go